Since these are the characteristics for adaptive immune response, do you remember the general characteristics for the innate immune response? Anyone? General characteristics of innate immune response. We have just gone through it.
It is non-specific.
Okay, non-specific. What else?
Present in healthy individuals.
Very good. As long as you're healthy, it is present. Okay. All right. For adaptive immune response, the host defense is stimulated by presence of microbes in tissues, and the microbe must have invaded the tissue. All right. So although it is more effective in combating the pathogens, it has its own terms and rules and regulations, terms to be fulfilled. This immune response can adapt to the presence of microbial invaders, but it's not the first line defense mechanism. Okay. As we know, the key players for adaptive immune response is your B and your T cells. Okay, the B cells will see the soluble antigens and the antibody will be released. All right. So with that, elimination of the microbe happens. I think we have gone through this earlier. But the T cells, they need to see the antigen on the surface of cells, meaning the pieces of antigen will be presented in molecules called the MHC molecule with the help of APCs, antigen presenting cell. So the antigen presenting cell, for example, macrophage and dendritic cell, they will take up the antigen, it is processed or digested. The pieces of antigen will get caught in MHC molecules. Okay, so for T cells to recognize the antigens, it needs the help of MHC and APC. Your APC can be your dendritic cells and your macrophages. T cells. Anyone knows what is CD4 positive? T cells? Referring to which cell? CD4 T cells, referring to, or another name?
Are they the helper T helper?
A guess? Very good. T helper cells. CD4 positive cells are actually T helper cells. Okay. CD represents a cluster of differentiation. Okay, these are all surface proteins. So if the surface protein is present only on a particular cell, all right, that particular surface protein can be used as a marker or to tag the population of cell. For example, here, if the CD4 protein, surface protein is only expressed on T cells, then we call it CD4 positive T cells. And that can be used as a marker to identify this population. For example, if you withdraw blood from an individual, and then you would want to analyze the population of CD4 positive T cells only or T helper cell only. So tagging will be done, or identification of that population will be done by using the CD4 as a marker. The T cells which can bind to this surface protein, all right, when the expression for this CD4 is positive, those population is actually T helper cells. So these are usually used for labeling or markers. So CD4 positive cells are T helper cells. Okay, so the T helper cells will see antigens in MHC molecules. They will proliferate or increase in number. They secrete help factors here, simply referring to cytokines. Okay, example of cytokines, you have interleukins, interleukin 2, 4, 6, also known as ILs, IL2, IL4, IL6, IL5. Some of the examples of interleukins released by T helper cells. So those kinds, or in this case, factors can help the B cells and CD8 cells. Each of these cytokines, it has its own function. For example, IL6, known as a pleiotropic function, meaning it has wide variety of functions. Okay, So what you can do is you can do a little bit of reading on the functions of each of these interleukins. So usually for interleukin 2, it can regulate the activity of white blood cells and responsible for general immunity. Okay, so that would help you in terms of enhancing the function of other immune cells as well. All right, next is your CD8 positive T cells, also known as cytokine cells. You can take these subsets of your T cells. They are subsets. There are other populations of T cells. Okay, CD8 T cells are known as cytotoxic T cells or T cells. It can perform direct killing of the pathogen. CD8 T cells will see the antigen in MHC1 molecules on infected cells. They can get help from T helper cells. They proliferate infected cells. There's one more population. All right, these are the two common populations or subsets of T cells. There's one more subset of T cells. If you want, you can take note on that. It's called the regulatory T cells, or known as Tregs. Regulatory T cells, also known as Tregs. The labeling or tagging of that set would be CD4 positive, CD25 positive. You have double marker for that to confirm that is Treg. So these are because these are the surface protein expressed by that immune cell. Okay, you have CD4 positive, CD25 positive. That is known as T regulatory cell. Main function is to suppress the immune system. Now, if your immune response and your immune cells are activated to confer protection, what is this immune suppressive cell doing here? Why do you think Tregs are necessary, being an immune suppressive cell? What do you think about it?
Could it be because we want to stop the inflammatory reaction?
Okay, stop the inflammatory reaction. Okay, generally in healthy individual, the Tregs will help to provide the homeostasis in terms of a balancing act. You see, when your immune system is overactivated, it can also cause, in a healthy individual, it can also cause autoimmune disorders, right? So you need Tregs to provide the balance. How if Treg, as I told you earlier, we can study the population of these cells, for example, the blood from an individual, and then you do this tagging just to isolate certain population, like you want to study on helper, you want to see what is the percentage of T helper, and then you want to see the percentage of cytotoxic T. So you have this tagging CD4, CD8 as a marker. Let's say in a patient, cancer patient, Treg population is high in number. What could that result in? Let's say Tregs, you know the function. If the Tregs are high in number in a cancer patient, what can happen? Just think about it, correlate with the function. Anyone want to give a try?
Is it possible that the T cells will also attack the healthy tissue?
All right, any other try? Good try. Anyone else? What can happen if population of T regulatory cells are high in a cancer patient? If you look at the function of regulatory T cells, it's an immune suppressive cell. So when the population is high, what will happen is that it will suppress the immune system. Anti-cancer activity will also be suppressed. Am I right? Indirectly, it can enhance or promote tumor development. Is that okay? Get that? Any response? When the number of B cells are in your system, particularly we are referring to a cancer patient, that's an example that I gave you, right? Mechanism of anti-cancer activities will also be suppressed, right? Because the function of Treg is to suppress the immune system. So when the anti-cancer activities are suppressed, it can promote tumor development. So it can actually worsen the scenario. All right, so that's all about Tregs. Okay, probably we have more details about T helper cells. Helper T cells, they have receptors for recognizing the antigen. With an antigen, they release cytokines to stimulate B cell division, meaning they can also enhance the function of another immune cell. Okay, the T helper T cell is the key cell to signal an immune response. If helper T cells are disabled, for example, in this case, people with AIDS, the immune system will not respond at all. So you have a key player here, T helper particularly. Okay. T cells, in general, they'll produce antibodies upon activation. Those with antibodies that bind with the invader's antigen are stimulated to reproduce rapidly, meaning as long as they are activated by specific antigens, they will increase in number. These cells will differentiate into either plasma cells or memory B cells. So once the B cells are activated, all right, they are known as plasma cells. They release antibody, and then there is another group, another set of cells known as the memory B cells. So the B cells can differentiate into plasma cells to release antibody, and also it will also differentiate into memory B cells. So plasma cells rapidly produce antibodies. The memory cells will retain the memory of the invader, and it will be ready to divide rapidly if an invasion occurs again. Okay, meaning invasion of similar pathogen occurs, the memory B cells will take place. Okay. Okay, this we have discussed earlier. What is self and non-self? This rule does not apply to autoimmune conditions. Everything that is encoded within the genome of that organism is actually self, and those that are not encoded within the genome is non-self. Okay. So we have spoken about this earlier. Non-self, example of non-self substances, harmful microorganisms, foreign proteins, transplant organs. Incompatible blood transfusion is also categorized under non-self. Okay, you have some terms here. I think you're very familiar with antigens, but you still need to know the definition. Substances that can trigger the adaptive arm of the immune system. All right. Example, bacteria, virus, fungus, and also parasites. Epitopes. This might be new to you. All right. Epitopes are areas exposed on antigens. You can only find this on antigens, not on immune cells. Okay. Epitopes are areas exposed on antigens that are recognized by antibodies and lymphocyte receptors, for example. So they're also known as antigenic determinants. All right. And it's interesting to know that there could be more than one epitopes on an antigen. In this case, this is an antigen. It's an example, a representation actually. Okay, let's say this is an antigen. You can have more than one epitopes. In this case, there are 4. Okay, these are the possible epitopes. So this is the site where communication occurs between the receptors of the immune cells and the antigen. Okay, as for the antigen presentation, the cells of the immune system have been trained to recognize self-protein and non-self. If an antigen protein is encountered by a macrophage, it will bring the protein to a helper T cell for identification. And if the helper T cell recognizes the protein as non-self, it will launch an immune response. So we have touched on this. Virus-infected cells will release interferons to communicate with non-infected cells present on the virus. Okay, generally interferons are signaling molecules or signaling proteins. Okay, they will be released by the host cells. Okay. So we have a depiction here for adaptive immunity. We know that two key players are your B and T cells. The response by B cells, whereby it releases antibody, is known as humoral response. All right, and the response by your T cells are known as cell-mediated immunity. So basically, for major arm of the immune response, it will branch out to innate and adaptive. Under adaptive, there are more subsets: humoral and cell-mediated. Humoral and CMI, cell-mediated immunity. Humoral refers to B cell response, and cell-mediated immunity, CMI, refers to the T lymphocyte response. Okay. For example, here there's a rodent infected with infectious agents. Okay. As for the B cell here, you have the B cell receptors, as you could see. Once the antigen has been recognized as non-self, only the antigen selected antibody, all right, antigen selected antibody secreting B cells will secrete the antibody. What does that mean? We will look at it in the upcoming slides. Not necessarily all the B cells will get activated and then they start to release antibodies. No, we have a selection here. Okay, we will discuss that in the upcoming slides. But generally, the B cells will start secreting antibody to eliminate the antigen. As for the cell-mediated immunity, if you could see here, you have T cell receptors. This is for T helper, and this is for cytotoxic T or cytolytic T. In this case, it's mentioned as killer T. They are the same. So you have the T cell receptor here. Once the antigen is recognized as non-self, all right, if you could see here, direct killing is happening. All right, there's direct killing of infected cell. This is your T cell, and you have the direct killing of the infected cell. Here, if you could see your T helper cells, they release cytokines. I've given you the example of cytokines just now, IL-2, IL-4, IL-6, for example. And the general rule here, the T cell receptors can only recognize antigen presented on MHC molecule. So that is the general rule. Okay. We have gone through this just now. These are the conditions for activation of the adaptive immune response. Although it's very much specific, you need to fulfill these terms. You need to fulfill these conditions for the activation. Adaptive immunity is only triggered if the microbes or their antigen, number one, have passed through the epithelial barriers. Number two, the microbes have been delivered to lymphoid organs or tissues. And number three, the microbes are recognized by receptors on the lymphocytes. So these are the rules to be fulfilled. Okay, let's look at some examples of this. In this case, let's say a local, for example. Okay, this is supposed to be a microbe, right? There is adherence of microbe to the epithelium here. And then penetration of epithelium. So these have fulfilled the rule number one. Okay, penetration of epithelium. So now adaptive immunity can take charge. Next, antigen uptake by Langerhans cells in the skin. So they will uptake this antigen here, the red color dots. And then the Langerhans cells will leave the skin and enter the lymphatic system. So all these terms and conditions are being fulfilled. Okay, the Langerhans cells will leave the skin and they will enter the lymphatic system. And those cells will be taken to the nearest draining lymph node. If you could see here, they have uptaken the microbe, the red color dots. So the immune cells have uptaken the microbes and they are moving to the nearest draining lymph node here. Then here, recognized by lymphocytes that have antigen-specific receptors. Okay. You have your lymphocytes here. Right. So it will be recognized by lymphocytes that have antigen-specific receptors. The lymphocytes that have a specific receptor for this particular antigen. Okay. And it will activate the antigen-specific lymphocytes. So once recognition happens through antigen-specific receptors, that will activate antigen-specific lymphocytes. Okay, these are the antigen-specific lymphocytes. There could be various lymphocytes, but only these lymphocytes can react specifically to this particular antigen. So that is why it's known as antigen-specific lymphocytes. Okay. The activated antigen-specific lymphocytes will leave the lymph node. The job is done, so it will leave the lymph node. It will migrate back to the site of infection to eradicate the infection. So it's going back to the site of infection to eradicate the infection. So the whole process could happen because it has fulfilled the conditions by adaptive arm of the immune response. So that's more of a graphical representation. So as you know, when we talk about lymphocytes, generally they have receptors on cell surface. You need these to recognize specific antigens. For B lymphocytes, they have B cell receptors. For T lymphocytes, they have T cell receptors. Okay, so these receptors. Okay, so let's say this is a T lymphocyte, dark blue here, T lymphocytes. On the surface of these T lymphocytes, you have surface glycoproteins. Okay, you have surface proteins. Example, you have CD4 or CD8, surface protein. You have T cell receptors in general. So these are some of the examples. There are many more. So I've provided you some examples just to show that on the surface of these lymphocytes, there are proteins, cell surface proteins. So that is why it could also be used for tagging or as a marker to study the population. Again, for B lymphocytes, let's say this is the B lymphocytes, you have cell surface proteins, for example, CD40, CD19. Generally, of course, will have your B cell receptor, but there are also other proteins, surface proteins. Okay, this is just to show an example. When naive B cells encounter a specific antigen, not just an antigen, but specific antigen, clonal selection will take place. So we are moving towards understanding clonal selection. Okay, so this is all about clonal selection. Okay, now I do not think that it's very tricky. Okay, just understand the basic concept of this. Okay, I've provided sufficient data for semester 1. Okay. Okay, let's say you have B cell here. Okay, these are your B cells. You have your antigens. In this case, it's yellowish orange. All right. These are your antigens and these are your antibodies. Okay. The invading antigen will bind to antibodies on one B cell. So if you see here, this is the B cell, whereby the antigens are binding to it. The rest, no. So this is a specific B cell which has the antigen recognition for this particular antigen. So that is why it can bind here. So the B cell, the selected B cell by that antigen will multiply rapidly. So in this case, we could see the receptor is red in color. Only the B cells with red color receptors are being multiplied, if you could see. The rest, no, although they are B cells, because they are not specific to this particular antigen. Okay, and then a large amount of genetically identical B cells will be produced. They'll be genetically identical because only these B cells are specific to this antigen. And what is the point of proliferating all the other B cells which does not have a specific receptor for this antigen? It cannot act against it. So that is why we need clonal selection. Okay, so only the selected cells, the B cell, will increase in number. And then that B cell will differentiate into plasma cells and memory cells. So it has been simplified for your basic understanding. All right, so your depth is what is required for this particular subject. So for clonal selection, this has been simplified. Basically, the B cell that is specific to this antigen, only that B cell will be increasing in number. So what is being specific here is the receptor. These receptors are very specific to this antigen. Okay, that is all about clonal selection. There is no point in increasing the number of the cells. Perform antigen-specific defense. Okay. Okay, now we are moving towards the primary immune response. Okay, because we are touching on memory cells. So, what is your knowledge of primary immune response? What do you know about primary immune response, apart from what has been shared here? Anyone? Not very sure. Okay. Generally, when we talk about memory cells, they are produced if antigen-specific T cells are activated. They provide the immune system the ability to recall previous encounters. Okay. With that, it will result in a quicker and larger immune response. So that is the function of your memory cells. Okay. For second immune response, antigen for some time. All right. If memory cells have been produced in primary response, the lag phase will be very short, or maybe it's not present. The immune response is rapid and bigger. Okay, this is secondary immune response, meaning if an individual is encountering the same antigen again, that means the memory cells could still remember how to react to that antigen. That is why the immune response will be rapid and bigger. Let's look at the graph. This could be helpful. Probably any of you can try to summarize this graph. Right, for primary immune response. Can anyone summarize this graph? You have your time period here, days. And here it is the immune response. That's your graph, your X and Y. The immune response, and this is a time period for reaction for the immune response to take place. Anyone summarize? Just give a try, doesn't matter. Anyone wants to give a try? What are they trying to indicate in this graph about primary immune response? This is the first exposure to a particular, and then they would want to indicate how long it takes for that reaction of immune response to occur. Could anyone want to summarize, to give a try? Just as how you look at other graphs, you can summarize. Anyone wants to give a try?
So basically, is it showing the amount of time it takes for the immune response to develop? So basically, the elimination of the microbe.
Okay, good. So what is it about the time period? Shorter, longer?
Is it because like
Because they
the
have
time from 0
indicated
to
lag phase
7?
here.
Yeah, the lag phase, it means that this is the first time that the body has been
Sorry, the time
infected with that
consumed
microbe.
is?
From 0 to 7
Okay.
days,
Okay, very
it's
good. So you
showing.
have a time. Okay. Yep.
Yeah, I was saying from 0 to 7, the reason it's a long time is because it's the first time this body has been infected with that specific microbe.
Yeah. Very
So
good.
this is the reason it's a longer time, because they're
Okay.
making memory cells.
Yeah, very good. That's the concept actually for primary immune response, because it's the first exposure to that particular antigen. So what you have here is time period taken to provide the immune response here. So if you could see here, from 0 to day 7, there is a lag phase. Very slow immune response or reaction. It took some time here. After day 7, only then you could see the surge. So basically this happens because when there is first time or first exposure, your immune system will take up time to react, to recognize. So there will be a lag phase here. Lag phase indicates it is taking a longer time period to react. Infection by microbe X, and it is primary immune response, first time exposure to that particular microbe X. Yeah, we are trying to summarize that. During primary immune response, you have lag phase, simply because your immune system is taking time to recognize that particular antigen. It's the first exposure for the immune cell as well. Okay, comparatively, for your secondary immune response, anything that you would want to, anything that you guys would want to summarize? Want to give a try?
The secondary immune response is much faster because there's already memory cells that are in the body.
Because there are memory cells. Okay, that is in general. But if looking back at the graph, how would you want to summarize this?
There's no lag phase.
Very good. There's no lag phase at all. Where? First part of the graph, infection with microbe X or reinfection of microbe X?
Reinfection, there's no lag phase.
All right. So that is because of memory cells, is it?
Yeah.
Okay. My question would be, as you have your B and T cells, T cells mainly, when they are activated, they will differentiate into the effector T cells, which is activated form of T cells. And the other part, you have the memory cells. Am I right? Memory cells, do they take part in the current elimination of pathogen, or they would only be active during the reinfection? Do you think the memory cells will take part in the current elimination of pathogen?
Only in reinfection, right?
Only in reinfection. Anyone else with a different answer? Do you think memory cells will take part in the current infection or reinfection?
Only reinfection.
Only
Yes.
reinfection.
Infection.
All right. Generally, memory cells, they do not take part in the eradication of pathogen, the current eradication of pathogen. All right. They are more efficient and effective for reinfection. So if you look at this graph here, again, immune response, and then you have your days here. All right, number one, first one is for the reinfection part, there's no lag phase. Okay. Number two, if you could see the surge. For infection, if you could see the surge is until this level, for example. Surge of response. Here, the surge of response is until this level. Simply means for reinfection, no lag phase simply means that it's faster. Immune response is faster. And the surge is bigger, rapid. So with this, it can perform more effective protection. Okay. Anything else about this graph that you would want to know?
Doctor?
Yeah.
Yeah, isn't the proof that it's a faster immune response in the length of the x-axis? Like what's the explanation for the high curve in the immune response? Is it the amount of memory cells?
Sorry, can you repeat? Firstly, I can't hear you clearly. I don't know why.
Oh,
You
I
want
was
to know
saying,
why is there a surge, is it?
yeah, what's the reason for the very high curve in the reinfection.
If you could see here, the memory cells have already seen the microbe X earlier, right? And they already know how to combat it in terms of creating a specific response. Okay, the presence of these memory cells makes the response quicker and also higher response. Okay, because it has faced the microbe X.
The immune response in the y-axis, is it measuring the amount of memory cells, or what is it measuring exactly? Because the speed
It's
of the
actually it's measuring the response, whether you have a higher response or lower response. So when you have more memory cells, the response will be higher.
okay.
Yeah. Okay. Anything else on this infection and reinfection? Anything else on this? All right. If there are no more questions, then we can move on to the next slide. This is a general overview. All right. So let's see. So here, this is the time after antigen exposure. All right. We also need to look at the different phases. First, you have recognition phase. Next is the activation phase. Effector phase. You have decline phase, homeostasis to provide the balance, and the last part is your memory. All right. So to summarize the adaptive immunity that we have just learned, when your naive B and naive T cells, naive means they have not encountered antigen before. So when your naive B cells and naive T cells encounters the pathogen and it has been recognized as non-self, they will get activated. B cells releases antibody and increase in number. As for T cells, antigen recognition occurs with the help of antigen presenting cells and MHC. So when it is confirmed that there is a non-self substance, it will get activated, increase in number. So it will perform either direct killing through CD8 or to enhance the function of another immune cell by CD4. All right. So the part where it increases in number is known as clonal expansion. But as I told you, only the selected cells, let it be B or T, only the selected cell with a specific receptor for that antigen will increase in number. So that is clonal expansion. Okay. The activated cells will differentiate into antibody producing cell if it is a B cell, effector T cell if we are talking about activation of T cells. Okay, that's your activation phase. So your activation phase is here. That's where you can see the surge of reaction. All right, that is the activation phase. Next is the effector phase, whereby you need to eliminate your pathogen or your antigens. Okay. Humoral immunity refers to your B cells releasing antibody. Cell-mediated immunity or CMI refers to your T cells. Generally, there are 3 subsets. But for antigen elimination, the important 2 subtype of T cells would be your T helper and cytotoxic T. Okay, so this is it carries out the effector function. If the term effector T cell simply means activated T cells. Okay, you just have to remember that. And this is the phase, and this is the part where I mentioned to you, once the job is done, the particular cells have to be eliminated. The particular immune cells have to be eliminated. That is the decline phase or homeostasis. It's supposed to provide the balance. We do not want the activated immune cells to keep on surveying the system after elimination of the antigens. Okay, so they have to undergo apoptosis. And for the memory phase, you will have surviving memory cells which did not take part in the current ongoing elimination process. They are important for secondary response if they encounter the similar antigen again. That's where and when they will react. So this is the part where the surviving memory cells. Alright, so in total, these are the different phases to eradicate your antigen by your adaptive arm of the immune response, mainly by B and T cells. So that is the general summary for adaptive immunity. Yeah. Anything else on this?
Doctor, I had a question regarding the MHC cells,
Yeah.
regarding their function. So I just want to clarify that the MHC cells are responsible to present the antigens on the pathogen, right?
Antigen presenting cells will present with the help of MHC. All right. Okay. Yeah. Continue.
No, I just wanted to clarify the function of
Hello.
MHC. Hello?
All right. You have. Hello. I can't hear you.
Yes, ma'am. Can you hear me?
Hello.
Doctor, I'm putting the
Yeah,
question
I
in
can
chat.
hear you now. I can hear you
Okay.
now.
Okay. I just wanted to clarify the function of the MHC. Yeah, could you just do that for me? Because I'm not too sure about what the function is.
Okay, now we have MHC class 1 and also MHC class 2. All right. Can I request other students as well to take part? Function of MHC class 1 and MHC class 2. Can you make it as a discussion? The others, would you want to give a try? Anyone knows function of MHC class 1 and MHC? Or maybe we can start by saying MHC class 1 is presented on which type of cell and MHC class 2 is presented on which type of cell? Anyone knows?
MHC class 1 is for CD8 and MHC class 2 is for CD4.
Okay, that has been discussed in our slides as well. So, anything other than that you know of? Anything other than that? What is the difference between MHC class 1 and MHC class 2? Still have, we can discuss.
Is MHC
Function of
class 1
MHC class 1, anyone? Sorry, MHC class 1. We can't hear you. Can anyone else give a try? MHC class 1. That's the function. All right, MSC class 1, they will display the fragments of proteins from the cell to cytotoxic T cells. All right, this will trigger immediate immune response. Fine. MSC class 2, anyone want to give a try? Generally, the major difference would be MSC class 1, they will present the endogenous antigens. All right, MSC class 2, they will present the exogenous antigens. All right. There are more differences between these two. MSC is also known as HLA, human leukocyte antigens. The others? Can anyone else give a try also regarding MSC class 1 and MSC class 2? Anything else that you know of? Dania, any other differences that you know about MSC class 1 and MSC class 2?
I'm not sure which one is which, but isn't one of them for CD4 and one for CD8?
Okay, which is for CD4 and which is for CD8?
I think CD4 is class 2,
Okay, good.
and CD8 is class 1.
All right, any other differences about MHC molecules? So what do you know about endogenous antigens and exogenous antigens? MSC class 1, they present endogenous antigens. MSC class 2, they represent exogenous antigens. Okay. You're right, MSC 2 for CD4, correct. What else do you know about MSC? Generally, what do you know about MHC molecules or HLA molecule? I mean, I believe that this is not only learned under immune system, but generally you do have some basic understanding about major histocompatibility complex, right?
Do they code for cell surface
All
protein?
right. Very good. All right. Any other differences between MHC class 1 and MHC class 2? So generally, whether it's MHC class 1 or 2, general function of MHC, they will bind the peptide fragments from the pathogens and display them on the cell surface for recognition of the T cells. All right, so in this case, antigen presenting towards the CD4 cells, CD4 T cells are mainly done by MHC class 2. Okay, so other things that you also try to learn and understand would be major differences between MHC class 1 and class 2. Any other major differences apart from antigen presentation?
MHC, the antigen binding cleft is formed by the alpha 1 and alpha 2 domains, and for
Okay,
MHC2, it's formed by the alpha 1 and beta 1 domains.
okay, these are major differences in terms of features component. Right, very well done. So for the first question, present differences between MSc class 1 and MSc class 2. For the person who asked this question, are you clear about MSc class 1 and MSc class 2 in terms of the role in antigen presentation?
Yes, doctor. It's
Okay,
clear.
good. So MSc class 1 are mainly found on nucleated body cells, but for MSc class 2, they are mainly found on macrophages, dendritic cells, for example. So these are some of the differences for MSc class 1 and class 2. Okay. Right. Okay. Let's look at the quiz here. Describe the three conditions for the activation of adaptive arm of the immune system. Describe the three conditions for the activation of adaptive arm of the immune system. I know that if you could scroll up and you could check your slides, but I would appreciate if you could do it spontaneously without referring back to the slides. Three conditions for the activation of adaptive immune system. Quite a lot of background noise. If you need to mute your speakers, please do so. I appreciate that. All right. Describe the three conditions for the activation of adaptive arm of the system. Anyone?
So one is that the pathogen should breach the epithelial barrier, right?
Okay, very good. Very good. The other two conditions?
The lymphocyte receptors need to recognize it.
Okay. We need three conditions. Try to recall.
The antigens need to be brought to lymph nodes or tissue, was it?
Okay. Overall, I think the understanding is definitely there. All right. Okay, that is all for that's the quiz for this particular lecture. That's all from my side. Is there anything else that you would want to discuss?
Doctor?
Yep.
Just now, the
Yep.
three conditions that for activation, do they have to happen in that exact order?
Sorry, what was that?
Yeah, the three conditions for
repeat the question.
activation. Yeah, the three conditions for activation, do they always happen in that exact order?
Did they always?
Do they always
I
happen
really
in
cannot
that?
hear you.
Do
you
those
mind
three?
typing it in the chat?
Okay.
Yeah, that'd be helpful. Do the three conditions for activation always happen in that exact order? This is the condition for activation of the adaptive arm of the immune. Yeah. And the answer is yes. All right. Yes, Dania, did I answer your question?
Yeah, thank you.
All right. Yep. That's all about it for the adaptive arm of the immune response, actually. I believe for your upper semesters, there will also be overlapping regions because you also have lectures on innate and adaptive arm of the immune response. So the depth would be slightly different, but you have your basics here, which could be further enhanced later in the upcoming semesters. All right, that's all from me. Thank you very much.
Thank you, doctor.
Thank you, doctor.
Thank you, doctor.
Thank you, doctor.
Thank you, guys. Have a good day. All right.
Thank you, doctor.
You too, doctor.
Thank you.